Good day, today we will be learning about DNA and the code of life. There are two nucleic acids in this topic, and they are deoxyribonucleic acid or DNA for short and ribonucleic acid or RNA for short. These acids are made up of smaller units called nucleotide, and each nucleotide is made up of nitrogenous base, a sugar portion which in DNA is deoxyribose and in RNA is ribose, and lastly a phosphate portion. There are three types of DNA and they all depend on their location. Nuclear DNA is located in the nucleus, mitochondrial DNA is located in the mitochondria, chloroplast DNA is located in the chloroplast. Nuclear DNA controls the synthesis of protein, more on that later. Nuclear DNA also transmits characteristics from parents to offspring. We mentioned earlier that DNA is made up of sugar, which is deoxyribose, phosphate portion and nitrogenous base. There are also two groups called purines and pyrimidine. Purine are adenine and guanine, and pyrimidine are cytosine and thymide, and this is how everything connects. Cytosine pairs with guanine and adenine pairs off with thymide. It is important to note that no one has actually seen DNA and RNA. We only know about this model, which was put forward by the scientist Watson and Crick. In other words, what we studied so far is only a theory on what DNA looks like. Now a little history lesson. James Watson and Francis Crick teamed up to do research at Cambridge University. Meanwhile, at King's College London, two scientists, Rosalind Franklin and her assistant, were working on a structure for DNA. Rosalind Franklin realized that DNA was a double helix from the X-rays she took, but her assistant did not like her. He showed these X-rays to James Watson and helped him in his research, and the two later, along with the assistant, won the Nobel Prize, while Rosalind Franklin died of cancer in 1958. We learn about DNA. Now let's learn about ribose. There are three types of ribose and they depend on their location. Ribosomal, which is found in the cytoplasm of the cell, messenger RNA, which is found in the nucleus but moves out to the cytoplasm. Transfer RNA, which is also found in the cytoplasm. These all play an important part in protein synthesis. DNA is a single-stranded structure and the sugar in it is ribose. In RNA the thymine is replaced with uracil. These two are similar. They both have sugar with phosphate. They both contain nitrogenous base, adenine, guanine and cytosine, and they both play a role in protein synthesis. But they differ. DNA is only found in the nucleus while RNA can be found in both nucleus and the cytoplasm. DNA is long while RNA is short. DNA is a double helix while RNA is a single-stranded helix. One of the main goals for DNA and RNA is protein synthesis. There are three steps in protein synthesis. Transcription, movement of mRNA out of the nucleus, and translation. Transcription starts, the DNA double helix unwinds, then the weak hydrogen bonds of the DNA break. This forms two single strands of DNA. One strand acts as a template. This forms a complementary strand, which is mRNA, using free RNA nucleotides from the nucleoplasm. 3 base on mRNA made up a code which code for the amino acids. Second step. Movement of mRNA out of the nucleus. mRNA move out of the nucleus, through the nucleus pore, into the cytoplasm, where it attaches to the ribosome. The last step. According to the codons of mRNA, tRNA molecules with matching anticodons, they bring the required amino acids to the ribose. The amino acids link together by peptide bonds and form the required protein. Cataract. A condition caused by the lens of the eye becoming cloudy. Autonomic sympathetic. The part of the nervous system that responds when a person gets fright. Apical dominance. A phenomenon where the main central stem displays more growth than the side stems, at the tip of a tree. Altricial. A type of development where young birds are born with their eyes closed and no feathers. Yolk. The structure that provides nutrients to the developing embryo in the ovoviviparous. Cytokinesis. The division of the cytoplasm during the process of meiosis. Translation. The stage of protein synthesis that occurs in the cytoplasm. Sperm cells. Cells formed in males as a result of meiosis. Nucleotides. Monomers of nucleic acid. Karyotype. The visible representation of a complete set of chromosomes in a cell. Biotechnology. The use of living systems and organisms to develop or make products that help improve our lives. Ovulation. The release of an ovum from the ovary. Synapse. The microscopic gap between two consecutive neurons. Gestation. The period of development of the fetus in the uterus. Tropism. The growth movement of a plant in response to acid mules. Precocial development. The type of development in birds where birds are born with their eyes open and can feed themselves. Allantois. The extra embryo membrane that is responsible for the excretion of waste in an amniotic egg. Seminiferous tubules. Tubules in the tests where spermatogenesis occurs. Abscisic acid. The plant hormone that brings about seed doming when conditions are unfavorable. Phylogenetic tree. A diagrammatic representation showing possible evolutionary relationships between different species. Peptide bond. Bond found between the amino acids. Punctuated equilibrium. The theory that describes evolution as consisting of long periods of little to no change altering with short periods of rapid change. Homologous structure. Similar structure that performs different functions in the different organisms. Artificial breeding. The breeding of plants and animals by humans for desired characteristics. Codominance. The type of dominance where both alleles of a gene are expressed in the phenotype in the heterozygous condition.